Hello, welcome to everybody joining this webinar. We really appreciate your presence. Today I have the honor and the pleasure to have an interview with Dr. Jeremie Gras from the IPG in Gosselies, Belgium. IPG, the Institute of Pathology and Genetics. And I'm very happy that Dr. Gras is willing to have this session with us. Dr. Gras is trained as a medical doctor and a certified specialist in laboratory medicine, but also a very well-renowned expert in quality control for clinical laboratory medicine. If you would have a question during our interview, please do not hesitate to put it in the Q&A box. The Q&A box is at the bottom of your screen. It's not a chat, it's a specific Q&A box. And after the interview, then Dr. Gras and myself will come back to you to answer your questions. But with this, first of all, Dr. Gras, thank you very much also to you for your flexibility and willingness to do this interview with us in a virtual way today. As a first introductory question, I wanted to ask you, could you give the viewers some more perspectives and background on the organization IPG and also on your specific role within the organization? Thank you.
So thank you, Peter, and thanks Bio-Rad for the organization of this webinar. Thanks also to all the attendees that are here today participating to this webinar. So IPG stands for the Institute for Pathology and Genetics. It's a diagnostic center located in Gosselies in Belgium. We have a big pathology department, meaning anatomic pathology. We have a center for human genetics with plenty of geneticists working there. We are, of course, performing a lot of molecular diagnostics such as qPCR, digital droplet PCR, next generation sequencing. And I have the chance to be since early this year the medical director of the organization or the chief medical officer. Before working at IPG, I've been working in a clinical laboratory for 10 years. I started up a molecular diagnostics facility there from scratch. Performed the accreditation also for this facility, and I have experience of 4 years as a laboratory director. Thank you.
Very impressive. So thank you. And we are also very honored, like I said before, to have this interview with you, because it's certainly with COVID and all other things going on, the molecular aspects and the molecular tests are very important. Now, to come to my first question is, you wrote a book on quality control entitled Laboratory Quality Control and Patient Safety. Now, can you elaborate a little bit more on the aspect of patient safety and the link with quality control?
Yeah, so I wrote this book already a couple of years ago, and certainly I would say that quality control plays really a key role in ensuring the quality of the patient results and just assuring to be absolutely sure that the results that we give are correct. It's certainly important for all laboratory testing, but we've seen that with molecular diagnostics in complex times, such as in the pandemics with COVID-19, it's absolutely, it's really important that you ensure that the results that you are given to your patient, given also to the authorities, are correct. So I would say that in quality control management and principles, there was a shift a couple of years ago from ensuring the analytical safety towards reducing the patient risk. It was certainly a focus of two documents of the CLSI. The first one was EP23, published in 2011. And the same focus on reducing patient risk was kept in CLSI C24, the fourth revision of the document, for which I was part of the writing committee, and it was published in 2016.
In the book, you also describe quality control principles for different departments in the laboratory. And we know all that quality control is pretty well developed in the core lab, let's say, of a hospital or a private lab. But what's the situation nowadays in a molecular lab?
So I think it's a very good question. I would say that molecular diagnostic is not exactly the same as compared to the core laboratory, because people working in core laboratories with big automated chemistry, immunoassays, hematology instruments, I would say that quality control was developed on such kind of platforms. The traditional works and publication from different authors really focused on immunoassays, clinical chemistry. So maybe it's a little bit a new concept or newer concept for molecular diagnostics. And also I would say that molecular diagnostics inclusion in a clinical laboratory is certainly more recent. So probably also the people working, the experts in molecular diagnostics, certainly they are scientific experts. Many of them, they are coming with MSCs or PhDs, and they are coming from the research world, which is a little bit different as compared to the classical, let's say, core laboratory. So probably I would say that quality control is less widespread, or the classical principles are less widespread in a molecular diagnostics laboratory, because it's a newer discipline and the experts in this field are coming from a different world where we know that quality assurance is sometimes an issue in research laboratories and in scientific publications.
So, if I understand you correctly, you say that certainly the principles and that you also described in the book could be transferred to the molecular lab, even though it's not so present at the moment. But what is then the main challenge in this context?
I think it's very important to mention that now with qPCR, you have quantitative values that you can use for quality control and quality management. So I think one of the first points is to know that even with qualitative testing, such as COVID-19 testing, even if you perform a qualitative PCR, you will get quantitative value. And the quantitative value that you will get is the CQ, the cycle quantification, so the point when the number of cycles when the PCR is becoming positive. It's a very important data that you can exploit. It's a quantitative value. You can plot it, you can play with it, you can follow the trend with it. So I think it's a very important point. As soon as you have a quantitative value that you can plot on charts, basically I would say that the traditional statistical quality control principles are applicable. So statistical QC is applicable to molecular testing, and you have with the CQ you have enough to do it. So as you have one quantitative value, the CQ, I would say what are the next challenges? Certainly I would say that training is certainly an issue, training the scientific experts, training the technologists also to the quality control principles, and yes, the requirements, the quality requirements of the big documents, either from CAP or either coming from the ISO 15189, which is mandatory since many years in Belgium, but also in France, also for molecular diagnostics. Very important also to follow the guidelines, and you have different guidelines coming from ISO, from national scientific bodies. You have also scientific articles that are discussing that. But certainly I think there is also many things that are still to be done regarding the application of traditional QC principles to quantitative PCR. If you look in the literature, the resource is limited, so I think there is plenty of room there in order to offer more guidance. Still, I think the principles can be applied to molecular diagnostics, but still there is plenty of work to be done to increase really the quality of what we do in molecular quality control.
Understanding the importance of the day-to-day and also the long-term follow-up of QC and also the traceability aspects, could you share with us an example where you take us through how you are following this, and maybe a Livy Jennings chart and some other aspects that the viewers could be interested in?
So traceability is a key component of any quality assurance system, not only in medical laboratories, but also in other type of businesses. So for quality control management in a molecular laboratory, it's very important that you have the traceability regarding this control. And traceability is not simple. It's which quality control material, perform on which extractor, which PCR robot, which PCR system, and it's been assayed by with which type of reagents, what type of reagent lot, who was the technologist responsible for performing this type of test. So all this information, you have to trace them. And how to do it? Of course, it's important to have one system that allows traceability of the operator, traceability of the corrective actions, and also to use a system that is protected. So I would say that just following your quality control results in an Excel sheet is not enough, because we see that in many laboratories. Okay, it's a good start because you can do some nice graphs with that. It's for sure, but sometimes the Excel sheet will not be protected and traceability can be an issue. So of course, with the Unity real-time software that is used since many years in clinical laboratories, in core laboratories and so on all over the world, you have a powerful tool to perform also your quality control management and full traceability for molecular diagnostics. I would say that in Belgium already since many years, probably already since 2010, many laboratories have been using Unity real-time with plenty of different instruments, and the exploitation of the QC, the general trends, is really performed on Unity, because you have this full traceability of the operators. All you can do with quality control regarding exploitation of the data, I think that there's no better program at the moment in the world, and I've been checking other programs, but until now it remains the one with the most functionalities, I would say. And you have a very robust system when you have an audit. We are in times where laboratories are going to be audited either by accreditation bodies or by the authorities, because they want to be sure that you are doing a good job with your COVID testing or other testing. It's a general trend, I think, in the healthcare sector that you need to have full transparency, traceability, and accountability for what you do. And I would say that I feel much more comfortable with Unity real-time and all my QC results into it that are backup, that are protected with the full traceability, that if I have only an Excel sheet. So Unity real-time is certainly a good solution, and we can show some practical examples. Okay, so this is a first slide here when I get this classical example of Chlamydia trachomatis result, QC result on a Levy-Jennings chart. So basically this is done using Bio-Rad independent controls, and we are just plotting the CQ values of the assay of the positive control here. So maybe it's a little small here, but you can see the mean, which is 24 point something. The mean is there. All these different points are representing a different QC value from a different day. And you can see all this variation there. Obviously, there was a problem here and probably here as well. So I think it's just showing you very, very interestingly what can be done with molecular testing on a Levy-Jennings chart. So it's a classical Levy-Jennings with the mean plus 1 SD, plus 2 SD, plus 3 SD, and minus 1, minus 2, minus 3 SD. So this next chart shows you the possibilities that we have in Unity to plot different types of Levy-Jennings chart. Here, I show you an example regarding Chlamydia trachomatis and Neisseria gonorrhoeae testing. Generally, this type of tests are run in multiplex PCR, either in a duplex or a triplex or even a quadruplex. And just to show you, on top you got the CT, and on the bottom you got the NG. So I think it's a very interesting feature with the combined view, because obviously you see that this kind of a multiplex testing shows a little bit the same behavior. If you look like here, probably we have a problem for both molecular targets. Here as well, so I think it's an interesting feature. Moving again to the next chart, just to tell you that even with molecular testing, you can work with the feature of the analytical goals or the quality requirements, which are typical, let's say, concept in classical QC, statistical QC for core laboratories. Using the user-defined features, you can define your analytical goals for molecular diagnostics. By example, here, 10%. And that's the view of the same graph for Chlamydia trachomatis with now the boundaries with analytical goals plus 10% and minus 10%. So it's a more advanced view of the Levy-Jennings chart. Again, you can see here that if previously with that point and probably with that point we were in infraction or out of control condition, if you were looking the classical Levy-Jennings chart with the plus 3 SD margin, here these points are still well within the analytical goals of 10%, which is quite narrow for molecular diagnostics test taken from extraction. Here is just to show you, okay, it's really, you can do pretty much whatever you want with the user-defined analytical goals. Here again is the same graph with the 25% analytical goal. So you can put also the graphs of the internal extraction control or the internal amplification control or simply the internal control monitoring. So this example is for the same multiplex assay, but here what is being charted on the Levy-Jennings graph, it's the internal control signal that is put into the negative control. So you can have an idea about, let's say, the inhibition picture of this PCR plate. Here the picture is a little bit different, but you can still monitor this signal as well. You can do it probably also by taking another type of control and spiking it with the internal extraction control to look at the behavior and to check your inhibition. Same with the analytical goals. And finally, you can combine all these different charts. So from the same assay, multiplex assay, you got the Chlamydia trachomatis, the Neisseria gonorrhoeae, you got the internal control, and you can check the general behavior of your different assays in your multiplex PCR. Also, we've been discussing quite a lot regarding the traceability, and this is a very important feature in Unity real-time. It's not just a chart tool or a Levy-Jennings tool. You can do plenty of other things with that, including reviewing all your QC data. So here it's a little bit small, but I have a close-up on the next slide. Okay, what is this saying? Here it's all the QC result coming from previous years. You get the reference laboratory number, the number of lot for the QC lot. You got the name of the assay. Here it's the Amplitrol from Bio-Rad. You got the instrument, you got the assay, you got the method used. You can even put the reagent traceability. You got the date and time. You got the value of the CQ value that was seen, the mean of the evaluation, standard deviation, Z value, the eventual QC rule, if it was accepted or not by room, if there is a corrective action, just to show you all you can do and all the different aspects that you have for traceability. And personally, I feel quite secure when I have this type of data to show to an auditor or to somebody from an official body. You can also check or use this application and this program to look at the QC rules violation or the QC data revision. Here, clearly, two samples for which the 1,4S QC rule was in infraction, and I think I have the details for this case later on. So this is an example of an out-of-control condition, again for Chlamydia trachomatis and Neisseria gonorrhoeae. You can see here that basically we have a problem. We have one assay that is spiking up here for the CQ value. It's for Neisseria gonorrhoeae and spiking down for the Chlamydia trachomatis. Clearly it's below 3 standard deviations here and above 4 standard deviations there. So clearly being seen. That's for the internal control. Yes, maybe I have to check back to this example, but it's simply a view with the analytical goals. And here you can see that clearly we are outside the analytical goals. I do not remember what was the boundary fixed in this case, but nevertheless we are certainly outside the quality specifications. Close to being outside specifications for the other molecular target for this assay. And just to show you the practical example of the PCR curves, here we are in the filter for Chlamydia trachomatis, and we can see that the fluorescence values are really low. It's 0.5, should be, normally this chart should go up to 20 or 25, so we are very low for Chlamydia trachomatis. But for this next slide, we see the signal for Neisseria gonorrhoeae. It's looking quite normal at first, but if you look at the fluorescence scale, it's really way too high. So maybe it's a message also always look at the curve, always look at the fluorescence value and also the CQ value, because you can also have an idea of something wrong that is going on by looking at these values. So in this case, clearly abnormal values for fluorescence for Neisseria gonorrhoeae, and we concluded to a contamination of the extraction system with a positive, high positive sample for Neisseria gonorrhoeae. What we have done is perform decontamination of the system, and we have run successful QC afterwards. So just to show you practical example coming from years ago, all what you can see, what you can find using Unity real-time with a full traceability and very interesting information showing that you can get practical value for practical problems that also can occur in molecular diagnostics with full traceability and really good security of the data.
To zoom in a little bit more in programs like Unity, the interlaboratory management, so the peer-to-peer comparison, is that something? I know in your previous lab in Bush you were using that a lot. Now in molecular lab context, is that something that also is growing? I suppose the peer groups are becoming also bigger and bigger. How do you see or work with that?
So it's a good question. Interlaboratory comparison is closely related to external quality assessment. So basically, if you look at ISO 15189, they speak also about interlaboratory comparison. And as you said, we've been using that a lot in many different places before. What's the advantage of interlaboratory comparison? If you take the classical core lab and if you compare it with EQA, with interlaboratory comparison using Unity, you have your internal quality control results performed by different operators in the daily routine that are going to be compared with a set of laboratories all over the world using the same instrumentation or the same platform or same methodology. And of course, it's very robust because you can compare your result to a set of tens of thousands of results that are coming from the everyday routine. So if you look and compare it to the external quality assessment, when you have 5 samples or 8 samples that are treated, like external quality assessment. Whoa, it's an EQA sample, be careful, check what you do, always performed by the same secretary, same technologist. It's not exactly the same realism, I would say. But that's for the core lab world. For molecular testing, I would say, certainly we can have this inter-laboratory comparison put into place because Unity is there, because we have independent quality control materials. But maybe this practice is not widespread at the moment, certainly. And I would say that probably it's a challenge because the market for molecular diagnostics is kind of diversified with plenty of different platforms. So probably it would be a challenge to define adequate peer groups and probably be a challenge as well to have quality control material with the same lot number that can be used for 1 year, 2 years, and have a big number of results so that you can compare your laboratory with. But I think that with the CQ value, with independent quality control materials, probably it's a good way to do it. It's certainly an interesting thing to perform in molecular diagnostics, even if it's a less widespread practice than in core laboratory.
Now, I wanted to come back to quality control in the core lab, or in, as you said, also the molecular lab nowadays. You hear a lot about third-party quality control and also the importance on similarity of quality control samples with the patient samples. Could you give some background or your perspective of this aspect and in the context of a molecular lab?
Yeah. So I think it's also an interesting question. With molecular testing, with qPCR, I think that we are in an area of more complex testing as compared to full automated testing in the core laboratory. So certainly because for qPCR, of course, we have now also big instruments, automated instruments that are coming, but I think that most of the big automated systems still rely on sample preparation, extraction for your DNA or RNA that you will put in your PCR, Preparation of the PCR plate, no, generally it's automated in 96 wells or 384 wells like we see now for the COVID. Then you got your PCR, then you will get your result. So we are in an area of a more complex testing with different phases. So I think that it's important with your quality control material that you are able to control all these different phases of testing. I think it's very important. And it's a general recommendation, especially in ISO 15189, that you work with quality control materials that you are going to test real like patient samples. So your quality control material for a good practice has to be extracted. Then you have to go to your PCR plate preparation with the quality control in the preparation of the PCR plate. Then you perform your PCR with your quality control material and have the control of all these different phases of testing. Regarding third party or independent quality control, it's an important question for molecular diagnostics, because now what you have is manufacturers that provide mainly quality controls that are included in the qPCR reagents. Or so you have your kit with your qPCR reagents and you have your quality control. So what is the problem? With this type of controls, you will only monitor the qPCR. But as we know, you can have plenty of problems in the extraction or in the PCR plate preparation. So working only with in-kit controls that only provide quality control for your qPCR, it can be problematic because you are not monitoring a big part of this complex phase of testing. In the global Belgian experience, plenty of non-conformities were given because for molecular testing, for a qPCR, for infectious disease, but also for hematology with tests such as JAK2 or BCR-ABL, plenty of non-conformities were given because you are not working with independent controls. And it's a recommendation in ISO 15189, it is in the chapter 5.6.2.2. So plenty of laboratories had to perform a corrective actions for that and find independent third-party controls for infectious molecular testing, but not only, also for oncology, molecular pathology, and hematology. So with in-kit controls, I do not know so many manufacturers that provides controls that can be used really as a patient sample. So if with the in-kit control you monitor only your qPCR, you have several problems. First, you will use it only in the PCR. You will not perform DNA or RNA extraction with that sample. So basically, it's not testing like you test a patient sample. Second problem, it's not independent. So with your ISO auditors, you will get a non-conformity. And then finally, of course, you do not monitor all the testing phase. So that's why I think that when you have independent third-party controls available for your qPCR or for your molecular assay, it's better to use them, of course.
So Dr. Gras, just coming back to quality control, we also have the EQA, the external quality assurance, quality controls. What's your perspective on this type of controls nowadays in molecular labs? Certainly now we have also the COVID pandemic, and how do you see that in molecular labs nowadays?
EQA, or external quality assessment, also called PT, proficiency testing, is also a mandatory part of the quality assurance program for molecular diagnostics. So it's mandatory, and as you know, also in ISO regulation, if you want to be accredited, you have to follow the regulations. And I would say that in many countries, even most countries, participation to a EQA, either organized by the country or organized by external provider, is mandatory. So you have to perform it, that's for sure. I'm always concerned with EQA with the limited number of samples that are provided in a EQA challenge or PT challenge. So generally you have 5, if you're lucky, maybe 8 samples. So you cannot rely only on external quality assessment. So to give an example, really an example for COVID, actual example, in our COVID-19 laboratory at IPG that we started. So it was a new laboratory that we started 4 months ago. We've been performing thousands of samples, even, more than 10,000 samples, and plenty of laboratories have been doing more than that as well with more automation. We've had 2 EQA challenges with a total of, if I remember right, 13 samples. So the question being, would you be secure if you perform 100,000 PCR in your lab for COVID? Would you feel secure with quality assurance with only 13 samples? Certainly, I would say that it's not enough. And of course, EQA is mandatory, it's an essential part of quality control for molecular diagnostic, but you have to design robust internal quality control principles and procedures in your laboratory so that you guarantee that the results that are coming outside of your COVID-19 laboratory or molecular laboratory are correct.
I understood that molecular testing is also very sensitive to contamination in the broader sense. How do you handle that in your lab, or how would you suggest to handle that as a potential risk in a molecular lab?
I would say that the issues or special issues for quality control in molecular diagnostics are certainly contamination, but also the inhibition phenomenon. So first, to discuss contamination, let's say physical separation of the rooms. It was one of the ways to control the contamination. Strict adherence to a pre-post PCR regulation or internal regulation or procedure was certainly something very important. But now we are in 2020. So we are working more with qPCR, for which you do not have to open your PCR plate or your tubes after an experiment. You have more automated systems coming from different manufacturers that are more automated, meaning you have your extraction, PCR preparation that can be done sometimes in the same room as your thermal cycler. So how do you manage contamination in 2020? Still, it's by strict adherence to the procedure, like decontamination of the robots, decontamination and maintenance, preventive or corrective maintenance for your extraction PCR extraction system. Also of your PIPTOR or PCR setup robot, it's very important to have a robust procedure with decontamination, preventive maintenance using special reagents or bleach or whatever. It's certainly important. Then, of course, because we are discussing quality control, it's mandatory, absolutely mandatory to work with a negative control. Recently, we had an example in our laboratory while we try to work with strict procedures, and we have a lot of experience with that. So it was, maybe a Friday evening, and I go to look at the PCR results, and you have 3 samples that are weak positive. What do? Do you validate these results? No. First, you look at your quality control results. And in that case, the negative control gave us slight positive signal with a CQ value that is certainly something that is very high. So CQ value of 38, indicating really a low positive value. And of course, we did not validate the result, and we performed the PCR again, even if it was late in the evening, or you can do it also during the night, but you have to relaunch these samples. Because in the context of COVID, what is happening if you release false positive results? You call the doctor, is going to call the patient, and is going to be quarantined. You are going to send the results to the authorities, and all the contact tracing chain is going to be put into place. The positive results are going to go into the epidemiology report. And then what's going to happen? Maybe your zone is going to be quarantined because you have a cluster of new cases. Of course, you cannot do that. But for extraction, PCR preparation, these are the instruments where you can have a contamination. And once you start with a contamination in the molecular lab, it's a nightmare because you have to swab everything to check where the contamination is. So you have to check with swabbing all the different parts of your system, like the bottles of reagents, and then the pipting system, and then the deck of the system. So strict adherence to a procedure with a good definition of the reagents to use to decontaminate the system and also use of a negative control. So that was for contamination, but we did not discuss also inhibition. So inhibition of PCR, it's a very important issue as well, because in complex matrices, by example stools, or again taking the COVID-19 example, pharyngeal swabs or nasopharyngeal swabs, these are also complex matrices, and sometimes you can have very difficult samples that can give you an inhibition. Inhibition is not completely known in PCR, but what's the risk of PCR inhibition is that, okay, you perform a PCR for COVID and you have no signal in the COVID reagent wells. Okay, can be a negative sample, but you have to check this phenomenon of inhibition. Generally, it is performed by using an internal amplification control. Can be targeting, let's say, a human housekeeping gene that can be present in the cells of the nasopharynx, or it can be also an internal control that is added during the extraction step, or generally during the extraction step or after the extraction step. And it's very important to look at that. When you have a negative sample, you should have a positive sample for your internal control. That is validating your qPCR result. So if you have a negative sample for the COVID mix, but a positive signal for the internal control for the same sample, it's okay. You have a negative sample and your PCR is not inhibited. In the contrary, if you have a negative result for the COVID mix and also a negative result for the internal control, then your sample is inhibited. And there, depending on your procedure and your laboratory, you can request a new sample, you can perform the analysis again, maybe with some adding of some specific reagent giving the problem that you can have, but it's certainly something that you have to do. Because also with the COVID, to give you an example, what's going to happen if you give a false negative result because you didn't check your internal control or you're not using an internal control. If you give a false negative result and the patient is stated negative, you send him back to work, you send him back to the family, you send him back to greeting the grandparents with the kids and so on. And that also can be a problem of developing clusters and so on. So again, controlling the inhibition is key. That is being done by using internal amplification control. And it's also a special issue in molecular diagnostics that makes it so interesting to discuss quality control for this complex testing.
Now, we discussed it already from different angles, but could you, in a kind of summary, could you give some key rules for a good quality control design in a molecular lab, as a kind of summary that you say, look, from your perspective, your experience as a medical director, if people want to start or already growing in their quality control, what are the key rules or recommendations?
Okay, so my first recommendation is that you have to define your quality control strategy for your molecular testing in one procedure. So it has to be written. You have to have this mandatory procedure, quality control for my molecular assays. Then it's your choice. You put all your molecular assays in the same procedure, or you make a separate procedure for each testing. My recommendation would be to have one single procedure for your molecular assays, or at least the assays of the same type, such as statistical quality control procedure for infectious disease testing, for microbiology qPCR, for virology qPCR. You can have one for hemato or molecular pathology. So very important, you start with this procedure. Then in this procedure, you'll have to reference what are the recommendations in your country, in your region, according to your maybe accreditation guideline, could be national or international, such as ISO 15189. You have to reference as well the articles you are going to use as a reference for your procedure. Then it starts certainly with defining the general principles and the phenomenon that you want to control, such as contamination, inhibition, and global performance of your molecular assays. Certainly defining the quality control materials that you are going to use, which positive control, which negative control, which types, which manufacturer. Be sure that your quality control materials can behave and be treated like patient samples, ideally third-party controls, independent controls that are going to be extracted, put in the PCR preparation robot and in your qPCR. You have to define what's the exploitation, what are also the criteria for valid controls and invalid controls, and what do you do when you have invalid controls? What do you do to be sure that your patient results are not going to be impacted by this? So you have to define, okay, I have an out of control condition. What is the definition of this out of control condition? And what do you do? We redo the experiment, we redo the testing since extraction, by example, or we invalidate the run and do something else, but you have to define what you have to do to be sure that no wrong patient result is going to come out of your laboratory. Then it's also, let's say, define the general principles on how you are going to define your protocol. Meaning, are you working with a specific analytical goal? By example, you do not want any CQ result to get out of your laboratory with more than 20% variation. You can set your analytical goal yourself. Define your QC frequency. Okay, I'm going to use 2 controls, 3 controls, 6 controls every PCR plate. Or it's the choice of the laboratory director to do that. And of course, it's also risk-benefit ratio. The cost analysis is also very important. But for sure, it's not the same doing quality control for a PCR plate where you have 20 samples or doing quality control for 384 wells PCR plate. It's not the same. Very important and specific for qPCR, you have to define the position of your quality control samples. It's a general recommendation that on your PCR plate, the QC samples should not be always at the same place. So it's not always A1 and A2. You have to modify the position of your quality control samples. Then, of course, what are you going to do with the results when they are out? How do you monitor them using Levy Jennings charts or using another type of chart? By example, Levy Jennings with analytical goals. Maybe if you want to be a little bit more advanced, you have to define the quality control rules that you are going to use. So what are the type of QC rule, the QC frequency, the type of QC material. I would say that's my general recommendation, and it has to be stated in one procedure. If you do not have a procedure for that, it's going to be complicated. And my recommendation would be as well to have a specific procedure and not to put the quality control procedure in another instruction for a specific test. So that would be my general recommendation.
Dr. Gras, thank you very much for this input, and I'm sure that the viewers online in the different topics we handle today can take some very interesting information to bring to their lab and bring to the day-to-day operations. So with this, I would like to thank you very much for your time, willingness to be with us here today in this virtual setup. We really appreciate your hospitality, and with this I would like to conclude the session and also ask our viewers online if you would have a question that comes up. Some of you we already see have put in questions in the Q&A box, but if you would have a question still, please go to the Q&A box. It's at the bottom of your screen and fill in the question. After some technical rearrangement, Dr. Gras and myself will come back to you, and we will go through the questions and try to give answers to as many as possible during the live session today. If we would not be able to answer your question live today, be sure we will come back to you in writing with a specific answer on your question. So thank you very much also to all of you who stayed with us today, and looking forward to talk to you in a minute for the Q&A. Thank you.
Thank you.